Research at Leeds is basically in 5 big research groups over all possible length scales. So we have astronomy and astrophysics, through polymer physics, through condensed matter physics, down to molecular and nanoscale physics, and quantum information. So we cover the whole spectrum of length in our research. All of those groups have excellent reputations, not just in the UK but internationally, and have PhD students both from the UK and Europe, but even wider in the world. Now, they're not separate groups in that there are significant collaborations between them, such as a biological overlap between the polymer group and the molecular and nanoscale physics group, and there's also interaction between the condensed matter research group and the quantum information research group.
My research is into NMR, which is nuclear magnetic resonance. This is the same physics that is behind MRI scanners that you find in hospitals. We use this to explore the dynamics and structures of polymeric systems, and this can be in biological samples and synthetic samples.
So one of the things I think that's a really exciting aspect of superconductivity is that it's an example of quantum mechanics operating at a macroscopic scale. So normally we think of quantum mechanics as being what controls things on a very, very small scale. It's what determines how an electron behaves in an individual atom. But in superconductivity, electrons throughout an entire sample, like be millimetres or centimetres, metres or even kilometres in scale, all start acting together in a single quantum mechanical state. That means electrons on one side of the sample behave exactly the same and are completely indistinguishable from the electrons on the other side of the sample, even though they're separated by really enormous distances. So this is why superconductors have no electrical resistance at low temperatures, because the electrons throughout the entire sample are all behaving exactly the same. Electrical resistance is associated with electrons changing direction or changing energy levels, but in a superconductor there is only one energy level for them to be in, and so they all behave exactly the same.
I'm an astrophysicist and I'm studying the life and death of stars. And sometimes people ask me, isn't that a little bit sad that you study dying stars? And then I explain to them that a dying star actually provides the seeds to build new stars. So actually it's quite a happy study as well.
Within the group, if I talk about the molecular and nanoscale physics group, we have a number of 5 academic staff, and most of those are new appointees, and they're all bringing very interesting and exciting new areas of research. So for example, Lorna Dugan is working on extremophiles. So this is how bacteria survive in extreme environments. And again, trying to extrapolate what we might learn from how a bacteria survive, we can start to maybe think, or how certain frogs, for example, also there are certain frogs that if you take to very cold temperatures, they effectively freeze, but they freeze without rupturing the cells within them. So you can then defreeze them and they can start hopping around again. And you know, obviously people would be very interested in if you could do this to humans, but there are big problems. You freeze a cell and it splits. So can we learn something about the way that the molecules or proteins within frogs and the cells within frogs are not ruptured or denatured as you take them to very cold temperatures, but they remain intact. And to do that, you need to understand about molecular interactions. To do that, you need to do very sophisticated physics experiments that involve neutron diffraction from proteins in solution. And so this is the type of work that Lorna's doing.
So my research falls under the bracket of soft matter, which deals with materials which are sort of spongy or gooey or runny, that sort of thing. So it's things like biological materials, blood, also pharmaceutical products, paint, and radioactive waste is a similar kind of thing. And all of these things are difficult to understand in that they behave in a more complicated way to traditional things like water, gases and that sort of thing. So partly for the industrial benefit and also just because they're theoretically interesting, there's this quite concerted effort to try and understand them in more detail.
The research I do is in the area of quantum physics, which is understanding the behavior of the universe on a very small length scale. So the scale of single atoms, single photons or molecules. And in this realm, things behave in a very strange way. We throw out all our common sense and logic about the world. So things can happen like particles can behave like waves, and waves like particles. Objects can pass straight through walls, and objects can be in 2 or more places at the same time. So it's a very strange world, but we can manipulate the behavior of these particles into lots of different interesting technologies, and that's what the field of quantum information is about.
So my research here at Leeds is based on looking at the chemistry within a disk around a young star. And the process of planet formation in these disks isn't very well understood. One of the theories about this planet formation involves the fragmentation of the disk. And when these disks fragment, they produce spiral waves, which we can image in different molecular lines. So my work here is involved making a chemical model of one of these disks with the hope that we can go away and observe this structure in the disk. And if we see it, then we can say, well, the conditions for this type of planet formation theory are happening, and this kind of lends support to that theory.
And the disks are major chemical factories. In fact, a lot of people have realized that the disks are important for producing prebiotic molecules. But the main interest that we have here at the moment is how do we use observations of the different molecules to figure out the dynamics that's occurring in the disk, and those dynamics are the ones that lead to star and planet formation.
Sarah Staneland gives a very different type of example of what biological systems might do, and she's working on something called magnetosomes, and these occur in bacteria. And the bacteria naturally, these particular types of bacteria, make nanoparticles, and these nanoparticles are magnetic. And so these potentially have application from all types of applications, from medical and healthcare to data storage at very high densities. One of the issues with them, though, is that they're not quite of the quality or the type of magnetic material that we would like for practical purposes. And so what Sarah has been trying to do is to not exactly genetically modify them, but actually certainly to modify the biological system so it makes new materials that they wouldn't naturally want to make, and with the properties that we would like to have in our system. So I think that just gives you a flavour of some of the types of work that are going on the biological interface.